Now you see, product of glycolysis. Pyruvic acid.
Sir, you only discuss up to aerobic respiration, sir. I think glycolysis is not started.
Yes.
Sir, I think glycolysis is not started. Sir, you will start today. Sir, you've only given the parts of aerobic respiration: glycolysis, Krebs cycle, and terminal.
This is the cytoplasm. It is an oxygen independent phase. Glucose, partially to form two molecules of pyruvic acid, two ATP, so this is what about the basic of glycolysis. Now I want to discuss here about the whole process. Now the first step, phosphorylation of substrate. What happens in this phase? I have to rub it out, please. Can I rub it out now? Can I rub it out now?
Yes, sir.
Okay, that's fine. First step, this is the first reaction. Here from ATP, one phosphate group is shifted to glucose. Whenever in any reaction ATP is involved or ATP is used up or ATP is produced, is actually the kinetic kinase reaction and has the enzyme name is kinase. Before kinase, you have to add the name of the substrate. Glucose is a hexose, whose the name of the enzyme is hexokinase. Hexose plus kinase, hexokinase. Clear? When in any reaction, either ATP is produced or ATP is used up, the enzyme is kinase. There is a kinase reaction actually. In any reaction, whether enzyme ATP is produced or ATP is used up, is actually a kinase reaction. And in kinase reaction, if you see substrate is a hexose, so it should be hexokinase. The next reaction isomerization. Glucose 6 phosphate glucose is converted into isomer fructose is phosphohexose isomerase. Phosphohexose because is not no more hexose only, as glucose 6 phosphate is phosphohexose, as isomerization occurs as the compound is converted to isomer, so it is phosphohexose isomerase. 3. Phosphorylation of substrate. Fructose 6 phosphate plus ATP. Fructose 1,6 bisphosphate. And there is phosphofructokinase. So whenever, remember this thing, whenever phosphate group is to be added to a compound, it was added to its last carbon. And when further addition of phosphate group occurs, it should be added to its first carbon. Are you getting me? Whenever phosphate group is added for the first time to a compound, it is added to its last carbon. In case of glucose, last carbon is in glucose last carbon is 6 carbon, so it is glucose 6 phosphate. Then it is from fructose 6 phosphate. When another phosphate group is required to be added, it should be added to its first carbon. That's why it is fructose 1,6 bisphosphate is produced. Then splitting. Fructose 1,6 bisphosphate is broken down to form 2 3-carbon compounds, and them is aldolase. Aldol reaction occurs, that is the carbon structure is broken down. CC bond is broken down, that's why it is aldolation. It occurs in absence of water, and fructose 1,6 bisphosphate is a 6-carbon compound. From it, a 3-carbon compound is released out, forming 2 3-carbon compounds. So this is what about the splitting reaction. After splitting, then isomerization. Hello? হ্যাঁ বলো। না না ক্লাস, আমি ক্লাসে আছি, ক্লাসে। হ্যাঁ ঠিক আছে, বিকেলে ফোন করো। একটু ব্যস্ত হয়, বিকেলে ফোন করে খেতে হবে। হ্যাঁ 4টার দিকে করো। Isomerization is, you see, is converted to the isomer It is triphosphate isomerase. Clear? Next, after that, now reaction start with 3-BGLD. Next step is phosphorylation and dehydrogenation.
স্যার সেকেন্ড স্টেপটা ওটা কি লেগেছে স্যার?
So here, oxidation of PGLD occurs, anionic gas reduced, enzyme is triphosphate dehydrogenase. Triphosphate dehydrogenase.
স্যার সেকেন্ড স্টেপটাতে এটা কি লেগেছে স্যার?
হ্যাঁ,
সেকেন্ড স্টেপটাতে
phosphorylation of substrate.
না না স্যার সেকেন্ড স্টেপটাতে গ্লুকোজ 6-ফসফেট তারপরে ওইটা
Phosphohexose isomerase is the enzyme forming fructose 6-phosphate. Go to the whole reaction first, then I will be repeating it again. Now, after once the DBGA is formed, the next step is what happens here? 1,3 DBGA. This is the first reaction where ATP is produced in glycolysis. Enzyme is again kinase. Diphosphoglycerokinase is the enzyme. This is a rare kinase reactions which is reversible. Generally, kinase reactions are found irreversible. Is a rare example of kinase reaction which is found reversible. 8. Shifting of phosphate group. What happens here? 3-PGA the phosphate group is shifted from third carbon to second carbon, forming 2-BGA. And the kind of mutation is the phosphoglyceromutase. After that, dehydration. Two PGA Manol compound is formed. 2-phosphoenol pyruvic acid. After that, Dephosphorylation. ADP.
স্যার একটি স্ক্রিনটা নিচে করবে।
Mucosa. Pyric acid plus ATP. Enzyme is pyruvate kinase. Look this one. Now I am discussing reactions one by one. First of all, you just go through these reactions. Now I am to suppose you know the differences. First of all, commonly use diphosphate and bisphosphate. Sometimes the term use diphosphate, sometimes bisphosphate. Why? Diphosphate and bisphosphate. Can you tell me? When the term diphosphate is used and when the term bisphosphate is used. Can you tell me?
When so there are more than one phosphate.
Group.
Yes, sir.
And bisphosphate?
I don't know.
দারুণ।
বিশ ফসফেটে দুইটা ফসফেট থাকে,
হ্যাঁ হ্যাঁ।
বিশ ফসফেটে দুইটা ফসফেট থাকে।
In both diphosphate and bisphosphate, there are two phosphates. Basically, in case of bisphosphate, they are attached to two different carbons. Then the term used is bisphosphate. Two phosphate groups are directly attached. Suppose A so here you see this is called bisphosphate. Bisphosphate, sorry. Diphosphate. You can call it adenosine diphosphate because two phosphate groups are directly attached to one another. Two phosphate groups are directly attached to one another. Then the term used is diphosphate. Clear?
Yes, sir.
But whenever the term bisphosphate is used, two phosphate groups are attached to different atoms. Suppose 1, 2, 3, 4, 5. One phosphate group is here, other is here. So you can call it bisphosphate. Clear now?
Yes, sir.
Okay. Now you see in glycolysis, glucose 6-phosphate. Now you see what happens here. There are two steps are called energy investment step. You need to remember this thing. Which two steps of glycolysis are called energy investment step. This is energy investment step. It is also energy investment step. Both these steps are energy investment step in the reaction number 1 and reaction number 3, because energy is invested in this reaction. Now you see, from reaction number 6, you need to double the product. From reaction number 6, you need to double the product. Double the product means, because once from reaction number 6, oxidation of 3-PGL is shown. Once it is completed, the DHAP is to be converted to 3-PGLD and the same reactions will be going on. So from here the product would be doubled. That's why in glycolysis 2 NADH, 2 pyruvic acid, 4 ATPs are produced, although 2 ATPs are used up, as net gain is 2 ATP. So remember this thing, from reaction number 6, the products will be double. Clear it is? Is it clear to you all?
Yes,
Yes,
sir.
sir.
Next, reaction number 6 is the oxidation step. Means this is the step in which oxidation of substrate occurs. This is the step in which oxidation of substrate occurs. So it is called oxidation step. Clear? Is it clear?
Yes, sir.
Okay. After oxidation step, then you see here, another most important thing. Whenever ATPs are produced in the 7th step and 10th step, ATPs are produced in 7th and 10th step, so ultimately we get 4 ATPs, and in both substrate level phosphorylation occurs. In both substrate level phosphorylation. Here also you get substrate level phosphorylation. দেখে নাও। Clear? Is it clear what is substrate level phosphorylation? When ATP is synthesized from substrate without involvement of electron transport chain, then it is called substrate level phosphorylation. When ATP is, write down, when ATP is synthesized from the substrate, when ATP is synthesized from the substrate without involvement of electron transport chain, that is called substrate level phosphorylation. Here in 2 steps, substrate level phosphorylation occurs. 1, when 1,3-DPGA is converted to 3-PGA, and last, when from phosphoenol pyruvic acid is formed. Is it clear now?
Yes, sir.
Now remember these things. What are the irreversible reactions is coming? Irreversible reaction of glycolysis. You need to remember these three. There are three reactions in glycolysis found which are irreversible. When glucose is converted to glucose 6-phosphate in the reaction number 1, and fructose 6-phosphate is converted to fructose 1,6-bisphosphate in reaction number 3, and when 2-phosphenol pyruvic acid is converted to pyruvic acid in reaction number 10. So these are three reactions which are actually found reversible, and the enzymes which catalyze reversible enzymes reactions are hexokinase, phosphofructokinase, and obviously lastly the pyruvate kinase. You need to remember these enzymes name, which are catalyzing irreversible reactions. Which are catalyzing irreversible reaction. Comes on some question comes. Name one kinase reaction which is reversible. Name one kinase reaction which is reversible, and this is number 7. Generally, kinase reactions are irreversible. This is the exception where, although it is a kinase reaction, but it is found reversible. Clear? Is it clear? Any doubt in any structure or anything? Can you? So what are the products of
Sir, reaction
glycolysis?
number 7 but wrong.
Huh?
Sir, এখন যে বলেছো reaction number 7
এক minute. Products, what are the products? 2 molecules of pyruvic acid, 2 molecules of NADH plus H plus, 2 ATP. But while writing 2 ATP, you need to remember these things. 2 ATP means actually Actually, 4 ATPs are produced. But two ATPs are used up. So net gain is 2 ATP. Clear? Now you can tell me your doubt.
Sir, step number 7 টা কি বলেছো reversible?
State number 7 হচ্ছে, you write down please. Kinase reactions, write down, are irreversible. Kinase reaction is irreversible. But diphosphoglycerokinase reaction is reversible. But diphosphoglycerokinase reaction is reversible. Comma, a rare example among kinase reactions. A rare example among kinase reactions. So you need to remember these things. It's a rare example among kinase reaction. Clear?
Yes, sir.
Okay.
স্যার একটু সাইড
You need to remember these reactions, all these 10 reactions. I think you can. You have to remember by these headings actually. Phosphorylation followed by isomerization, again phosphorylation, splitting, isomerization. These are oxidation step, phosphorylation and dehydrogenation. Then dephosphorylation, shifting, dehydration. Lastly, here one thing. You add water, forget it. In reaction number 9, when 2PGA is converted to TP2PEPA, a molecule of water is released out.
স্যার
বলো
স্যার একটা কথা ছিল স্যার।
হ্যাঁ বলো শিওর।
স্যার টাইমিং কি স্যার এখন 8টাতে ফিক্সড হয়ে গেছে?
হ্যাঁ 8টা না, আমি তো ভাবছি 7:50 করবো এটাকে।
না না স্যার কোনো প্রবলেম নেই স্যার, মানে 2 দিন স্যার মানে ক্লাস একটু মিস হয়ে গেছিল এর জন্য স্যার বলছি টাইমটা কি 8টা থেকে নিন।
টাইমটা এখন আমি 8টা না 7:50, অনেক সময় একটু দেরি হয়ে যায়, শীতকাল তো একটু দেরি হয়ে যায়, কিন্তু 7:50 আমি চাইছি আর কি এটাকে আলটিমেটলি পিস করতে।
আচ্ছা ঠিক আছে স্যার।
হয়তো কোনোদিন 8টা বাদ হয়ে যাচ্ছে, কোনোদিন 8টার চেষ্টা করবো, 8টার পরে 8টার মধ্যে মাস্ট আর কি। তাই মানে টাইম থাকবে 7:50, পারলে 7:45 এরকম করে একটা পিস করে নেবো আর কি।
ঠিক আছে স্যার।
অনেকেই স্কুল আছে এই জন্য আর কি।
Yes, sir.
You need to remember how many ATPs are produced in glycolysis. It's a general question. How many ATPs are produced in glycolysis? Generally, in anaerobic respiration, in case of anaerobic respiration, there is generation of 2 ATP. Basically, 2 ATPs are gained. But in case of aerobic type, anaerobic 2 ATP. Two plus 4 or 2 plus 6. 6 ATP or 8 ATP. Actually, the 2 NADH2, either it can give rise to 2 ATP or 4 ATP, or it can give rise to 6 ATP depending on the shuttle used. So that's why in aerobic respiration, either 6 ATP or 4 ATPs are produced, or 8 ATPs are produced, but in anaerobic type, only 2 ATPs are produced. Clear? Now, after that, pyruvic acid enters mitochondria. After that, pyruvic acid enters mitochondria. What happens here? reacts with TPP or thiamine pyrophosphate to form hydroxyethyl TPP. Lipoamide in oxidized form forming acetyl lipoamide. कोएंजाइम में lipoamide reduced, then plus FADH plus H plus. Lastly, NAD plus NADH plus H plus. So these are reaction carried out by different enzymes, but you know the products are you see ultimately these are the products. The enzyme together called pyruvate dehydrogenase complex. Just 3 minutes I am coming. Just 2 3 minutes. Okay. Understand? Just coming. In the first reaction, there is decarboxylase activity of pyruvate dehydrogenase enzyme. If I can rub it out like this. Decarboxylase activity of dehydrogenase. Pyruvate dehydrogenase. This is one. In the next reaction, dihydrolipoyl transacetylase. Here and here, these two reactions are catalyzed by dihydrolipoyl transacetylase. Then this one and this one catalyzed by dihydrolipoyl dehydrogenase. So this is a complex reaction, and hence you need not to remember this actually one in one, but you have to just try to understand this thing what happens, because I have to write here the reaction in nutshell. So what happens in this reaction, you see ultimately pyruvic acid plus this is a coenzyme forming This is the reaction. Enzyme is so why it is called complex, that's why I am showing this thing. Actually it is taking place in this way. In short, this is the reaction. Particular acid, gas dehydrogenated and decarboxylated to form acetyl coenzyme, which is a 2-carbon compound. Are you getting me? Here I am showing you. Can I rub it out? The lower side?
No sir, wait.
Huh?
No sir, no sir. Iktu.
Can I rub it out?
No sir, iktu.
Okay. Now what happens here? I am showing you. Pyruvic acid means CH3COC. So you see what happens from here. Irreversible actually. Irreversible. So this is pyruvic acid, this is the acetyl coenzyme. So this carboxylic group released out as carbon dioxide, NAD, gas reduced. So this is what about the reaction actually. Is it clear now? Is it clear? So this is the first reaction in aerobic respiration where carbon dioxide is produced. This is the first reaction in aerobic respiration where carbon dioxide is produced. Clear? Is it clear now?
Yes, sir.
Is it clear?
Yes sir.
Okay, that's fine. Can I rub it out now?
Yes sir.
Okay. After that, acetyl coenzyme enters the Krebs cycle. Actually, it is an intermediate step. Between glycolysis and Krebs cycle. Now Krebs cycle. The first reaction, the 4-carbon compound coenzyme irreversible reaction forming citric acid enzyme citrate synthase. Okay. একটু ক্লাস একটু ক্লাস আবার These are citric acid. This is citric acid. Clear? একটু দেখো হ্যাঁ গো বুঝতে হুম 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 ধন্যবাদ তো আগে যাবো না বুঝতে বুঝতে ঠিক আছে না লাগছে না এটা মুখলি কম লাগছে বুঝতে পারছি হ্যাঁ ভিটামিন নিয়ে দেবো আর একটু লাভ আছে ঠিক আছে হ্যালো আর কত একটা প্রবলেম ক্লাস একটু লাভ এই রিঅ্যাকশনটা বুঝতে পেরেছিস? ক্লিয়ার টু ইউ অল? That's why citric acid is again called tricarboxylic acid or TCA. Tricarboxylic acid or TCA. Clear? Or you can write TCA. This is the first stable product of Krebs cycle. But the structure is stable structure. It cannot donate hydrogen because you know, like likewise in Krebs cycle also, oxidation of substrate occurs by means of dehydrogenation. Oxidation of substrate occurs by means of dehydrogenation. So here also, what is important? The structure conformational change of citric acid is required to be taken place so that it can donate hydrogen to NAD. Can I rub it out? This is the first reaction of
Yes.
Krebs cycle in which oxaloacetic acid reacts with acetyl coenzyme to form citric acid. Oxaloacetic acid reacts with acetyl coenzyme to form citric acid. Clear?
Yes, sir.
Okay, that's fine. After that, the next reaction. With the liberation of one molecule of water, citric acid would become cis-aconitic acid. And you see, if you go to this reaction, this is the citric acid. When cis-aconitic is formed, it should be so this is the cis-aconitic acid, and obviously clear? After that, cisaconic acid plus H2 isocytric acid. What about the structure? Look here. CHOH. This will be CHOH COOH. So now this structure can donate hydrogen. This is called isocytric acid. This is called isocytric acid. After that, as hydrogen is removed out, isocytrate dehydrogenase. It appears like this, you see. No. CH2COOH remains same. Then you will get the CHCOOH remains same. Here this have to develop, so it is called COCOOH. This is called oxalosuccinic acid. But it is actually the most temporary compound. Before leaving the active site, it is to be converted to alpha-ketoglutarate acid. There's the same enzyme is catalyzing both. So the next reaction, what happens? Oxalosuccinic acid plus carbon dioxide. So here what happens? Enzyme is same. Isocytrate dehydrogenase. That means if you go to structure, one carboxylic group from here released to the carbon dioxide, so you get This is called alpha-ketoglutarate acid. And obviously the release of carbon dioxide. This is actually a unidirectional reaction. This is unidirectional reaction. Once it is formed, then alpha-ketoglutarate acid plus COSH forming carbon dioxide. So from here it gets oxidized again, and also decarboxylation occurs. It's called as alpha-ketoglutarate dehydrogenase. That means after that it would become these are succinyl coenzyme formed. That is the first 4-carbon compound produced in Krebs cycle. And this is the only 5-carbon compound found in Krebs cycle. Hello. After that, succinyl coenzyme plus NAD plus succinyl coenzyme succinyl thiokinase. Here, this GTP ATP is formed. So here also we'll get substrate level phosphorylation. Clear? Just a minute, coming. Then what about the reaction here? A structure. Suppose when succinic acid is formed, it should be nothing but this is the succinic acid. After that, I am rubbing out the upper part. Now, number 8. FAD plus succinate dehydrogenase. Reaction appear like this. What happens? From here, hydrogens are removed out, so CH3AA will get. After that, fumaric acid is broken down in the presence of water. Enzyme is fumarase, forming malic acid. The structure here you did the so what about the malic acid? Actually, water is added for this. Already water given. So here OH is added, and then you will get here CH2COOH. This is the malic acid. After that, in the last reaction, malic acid plus NAD plus malate dehydrogenase is the enzyme. Forming oxaloacetic acid. Plus and H plus H plus. So reaction label is CH so these two hydrogens are to be used up here to reduce oxidized malic acid, forming again we get the oxaloacetic acid. So this is the Krebs cycle. Why I am giving you the actually structure so that you can understand it properly. For proper understanding, actually I gave you the structures. Clear? Is it clear to you?
Yes, sir, clear.
Okay. Now the steps in which NAD gets reduced, the steps of glycolysis in which NAD gets reduced are, you know, in the step when isolatic acid is converted to oxalosuccinic acid, in the step when alpha-ketoglutarate acid is converted to succinyl coenzyme, and the step when malic acid is converted to oxaloacetic acid, and the step in which FAD gets reduced when succinic acid is converted to fumaric acid. So in three reactions in Krebs cycle, NAD gets reduced to form NADH plus H plus, and in one reaction FAD gets reduced to form FADH plus H plus. In two reactions CO2 produced when oxalosuccinic acid is converted to alpha-ketoglutarate acid, and again when alpha-ketoglutarate acid is converted to succinyl coenzyme. And two substances actually succinyl coenzyme. Is it given? Go through the reaction, please. If it is not given, write CO2. Alpha-ketoglutarate acid to react with NAD, COSH, forming succinyl coenzyme plus NADH plus carbon dioxide. Is this given?
Yes, sir.
Yes,
Huh?
sir.
Okay. So this is what about the Krebs cycle. And I think you know that you need to remember in which steps. So substitutable phosphorylation occurs only when succinyl coenzyme is converted to succinic acid. And all NADH, FADH are produced here are to be oxidized in the electron transport chain that occurs in the presence of oxygen in the inner mitochondrial membrane. Is it clear up to this?
Yes, sir. Clear.
Okay. So today I am to close here. In my next class, I am to start with the terminal respiration. And in the meantime, you need to go to the whole Krebs cycle. If you have any doubt, you please tell me. Clear?
Yes, sir.
Okay. That's all for today. Thank you.
These symbols in which both sides are there, arrows on both sides are there.
Actually, it means that this is this time. Reversible reaction.
Okay, sir.